This is a production of Cornell University.
Hello again, my name is Bahia Zal. I'm a second-year PhD student in plant breeding and genetics here at Cornell, studying hemp with Dr. Larry Smart. You guys probably know that today is our last webinar. We're closing this out. I just want to thank all of you for joining and participating in this series. It's been a really great experience, and I love all of the questions that we've received during the presentations, and I really think that that enriches the learning experience. Just as a heads up, FYI, our Cornell Hemp Field Days will be in September this year, with the Fiber and Grain Hemp event on Thursday the 12th, and then another event for the high cannabinoid hemp on Friday the 13th. To get news on these field days, you can go to the Cornell Hemp website and scroll down to the bottom. You'll see a little button that says "Stay in the Loop" on the right there, and this will sign you up for any hemp-related announcements. And the website link for that I'll put in the chat. Also, this webinar will be continued next year, so when that comes around the winter, be sure to register for that and join us again. Also, this last semester of the webinar series has been recorded, so you can share that with others and rewatch them. Those are hosted on our Cornell School of Integrative Plant Sciences YouTube channel. You can also, again, find more of those hemp resources on the Cornell Hemp website, and I'll include both of those links as a reminder. So thank you everyone for joining us. And with that, I'd like to welcome our speaker, Shelby Ellison. Dr. Ellison is an assistant professor in the Department of Plant and Agroecosystem Sciences at the University of Wisconsin-Madison. She's interested in the preservation, characterization, and utilization of genetic diversity in the development of new alternative crops, which will meet the needs of farmers and take into account environmental implications. She also has a focus on how humans interact with plants and how domestication and breeding have altered plant genomes. Dr. Ellison is also the lead for a nationwide feral hemp germplasm collection effort to rebuild our U.S. hemp seed bank, and is a member of the Midwestern Hemp Research Collaborative, which trials hemp cultivars for regional adaptation. Thank you, Shelby, for joining us today. I'll let you take it away with your presentation.
All right, thank you very much, Bahia. I'm super glad to be here. This webinar series has been excellent. I've been listening across the years and really enjoy it, and really honored to be a part of it this year. It is definitely feeling like summer in Wisconsin finally today, and classes end this week. And I know everybody is very excited to get out in the field and start the new cycle of 2024 hemp field trials and research. So it's good. Okay, so yes, today my presentation is going to be talking about feral hemp, and we're going to talk a lot about feral hemp and how we can potentially use this as a valuable resource in future hemp research. So without further ado, let's get all these ducks in a row. Okay, so an outline, I'm just going to talk briefly about the history of cannabis in the United States. I know many of you probably know more than this than I do, but it's going to set the stage for where feral hemp is coming from. Then we're going to talk about cannabis germplasm. Whenever I say germplasm, I just mean seeds in this case, and why it's important to preserve cannabis germplasm. Talk about a specific project that I'm a part of, collecting feral cannabis across the United States, and the important role of citizen scientists that have been helping with these collections. Then a little bit of the nitty-gritty of the research of what we have found so far and where we hope to head in the future. So a brief timeline of cannabis or hemp in the United States. So as many of us know, cannabis was brought over to the United States in the colonial era in New England in the early 1600s. At that time, it was primarily used as a source of textiles along with flax. It was kind of grown in some production, although flax was a main source of competition, but it kind of was used throughout the 1600s, 1700s, 1800s in low acreage. It spread across the country and kind of became centralized around the Midwest, specifically states like Kentucky and Missouri and Illinois grew a lot of hemp. And really one of the challenges associated with using hemp at that time was, as we all know, it's very difficult to process. So there were some key early 20th century inventions that led to the improvement of being able to break hemp for its fiber components. So here you can see one of these automated hemp breaks that was developed. And interestingly, so I'm coming from the University of Wisconsin-Madison. One of the reasons why Wisconsin became one of the larger growing regions of hemp at the turn of the 20th century is because for some reason we invested in a lot of hemp mills at this time and had these automatic hemp breaks that became very important for the efficiency of breaking hemp into bast and hurd fibers for other applications. And at this time, along with textiles, hemp was mostly used for cordage, and that's what the processing was important for. Then we all know that along this time period, we start to see the association with hemp and drug type cannabis being tied together, not being distinguished from each other. And there's racial discrimination associated with drug type cannabis. And because of this, we see things like the Marijuana Tax Act, which leads to high taxation, as well as the need to be kind of registered and licensed to grow all types of hemp. So this leads to a drastic decrease in the acreage produced. In the 1940s, we then know there is the Hemp for Victory program because during World War II, we see a shortage of hemp used for cordage, which is especially important for things like ropes and sail production. So the United States government encourages us to grow hemp. Peaking in 1943, we see this production here across the 30s and 40s where we have a tremendous amount of hemp grown for fiber in the 42 to 44 region. And this is really important for setting the stage for this talk because of this encouragement, because we see a ton of people growing hemp for the war effort at this time, this is ultimately going to be the source of a lot of the seed that escapes from production and becomes fertilized. So that's what I'm going to be talking about a little bit. And the regions that grew a lot of this hemp, specifically a lot of places across the Midwest, where a lot of these contracts took place, that's where a lot of the feral hemp escaped to. Then we know hemp and all types of cannabis are outright become impossible to grow with the Controlled Substances Act of 1970, where they are scheduled as a class 1. And this is obviously in the news right now with yesterday that there might be a rescheduling of cannabis coming forth. So I hope in our next seminar series next year, we're going to be talking a lot more about what the implications for that is in the hemp and cannabis world. So we stopped growing hemp. Now we're all here, right? So since 2014, hemp has become re-legalized to grow in the United States. So this is our brief history, focusing on this massive uptake of cannabis production that happened in the 1940s. So what are the things that need to happen now for the hemp industry to thrive? We've been hearing from people across the last couple of years, excellent speakers in all of these different areas, things that are really important. We need to have regulatory clarity and consistency across the federal and state regulation of hemp. What my big focus on for the rest of the talk is in research and development. So improved cultivar development, as well as product development, the infrastructure to make products, of course, as well as market demand and consumer education. I think this webinar series is excellent because, we can reach a lot of people to get the word out there about all the things that hemp can be used for and to further educate people on the differences between the different types of hemp and what the main bottlenecks are. Of course, sustainable environmental practices are key, I think, to the a resurgence of hemp cultivation, especially with lots of consumers being interested in the potential sustainability aspect of hemp as compared to other products, and as well as supply chain collaboration, which is going to be very important, I think, for the hemp industry to thrive. So I'm going to be focusing on the research part of this, and specifically about breeding in cannabis. So to be able to develop regionally adapted cannabis cultivars, so farmers are able to have high yielding cultivars that they can then make potential profit on, you need cannabis germplasm or seeds to kind of kickstart those breeding programs. So of course, this is important for crop breeding. It's also important to have a wide range of genetic diversity to be able to pull from to adapt to different climates. All in all, biodiversity conservation is very important just because, you know, big things happen, large shifts in interest, as well as we're talking about things like climate. Conserving biodiversity is of utmost importance, I think, for a lot of people. And then scientific research. So beyond crop breeding, there is lots of reasons why seeds are useful to study for the general physiological aspects of how these plants might thrive and other research applications. So the pinch point of this right now in the year 2024 is that because cannabis was illegal to grow in the United States, most of the seeds were either lost or they have been privately cultivated or kept. So most of the germplasm right now is owned by companies, or it is non-compliant, meaning that it has less than 0.3% THC. So if you're interested in developing new hemp varieties or cultivars, you are not able to use this material. So the goal then is, well, or the question is, what do we do? So how can we build up the new cannabis seed bank? So this is where my project comes along. And so growing up in Madison or in Wisconsin, I always was aware that there are plants that look a lot like, or I did what we call ditchweed, but now in the academic world called feral cannabis. So those escaped plants from when we were growing a lot of cannabis back in the 1940s. They persisted all throughout the landscape across Wisconsin, as I mentioned, most of the Midwest. And so this slide here, as we mentioned, there was a large uptick of this planting across the region. Essentially, these plants, a lot of cannabis never was fully domesticated and has traits like where the seed shatter is very common. So a lot of seeds during transportation would shatter into their local environments. So this is when, so the process of fertilization is when a domesticated plant then moves into the wild, and then it can slowly adapt through time to that environment, because there are random mutations that may lead to some sort of adaptation. So this is probably what has been happening across like roughly the last 100 years in the Midwest. And we know that these came from domesticated plants that were grown here that escaped into cultivation because there were not a true source of wild cannabis in the Midwest. It's not from the from North America. So these are true feral, endopheral populations. So this was an idea of, okay, we know these populations exist, so maybe we should collect them. So a project was developed between myself and Dr. George Weiblin at the University of Minnesota, who has been working in cannabis for the last 2 decades. And the idea was to collect all of this feral cannabis from around the region and get a better idea of, you know, is it actually compliant? What is the cannabinoid composition of these plants, as well as understand how much genetic diversity there is. So that might be utilized for breeding programs and understanding kind of how these populations come from different geographic locations. They have maybe become adapted to different climatic factors in those regions that can be used in breeding programs. So I'm going to be describing that project to you all now. So there's three main parts of the project I'm going to go through. The first is the actual efforts that we've made to collect seed from these feral populations, and how we've been selecting the places we've been collecting. So the main thing was that we wanted to try to collect from as many different ecoregions as possible. So an ecoregion just meaning that there are some sort of ecological factor that is making one region distinct from another. And you can think about these in terms of what those factors might be. So we know in cannabis, most cannabis is has some sort of photoperiod sensitivity. So if we look at this, there's other crops like soybean, which is being shown here, where you have different growing zones that are associated with the latitude or photoperiod of these regions. So collecting across a range of these different latitudes will relate to the photoperiod, which might make something more adapted based on its flowering time, whether we know that there's big differences between what's happening in Wisconsin and what's happening in North Carolina, and what the necessary photoperiod will be for maturity to have high yielding cultivars. We can look at this map of annual precipitation rate. You can see that there's a distribution of high rainfall to very dry regions. So you could imagine that plants that are existing in these regions will have different regional adaptation to perhaps things like water use efficiency that is necessary. So we wanted to collect across as many of these ecoregions as possible, but also considering that most of this feral hemp is distributed broadly across the Midwest. So how do we start to find populations? So as I mentioned, I knew of a couple in Wisconsin, but I didn't think that 2 or 3 from where I grew up was probably going to do the trick. So we turned to iNaturalist, which is a wonderful app that allows for you to, or allows for citizen scientists to populate biodiversity observations. So if you see something, you can take a photograph of it and log information. It could be anything from the GPS coordinates to if the flower is vegetative or reproductive, or if it any sort of feature about the flower or the plant. And this then you can filter based on genus or species, whatever you're looking for. And so if you go to iNaturalist and look up cannabis sativa, you'll actually see this map right here. So there are lots of records for cannabis sativa across the entire world. And then if you click on any one of these individual squares, it'll lead you to that observation, and it'll tell you the collector and when it was collected. Hopefully there will be an image or two there, as well as you can get the GPS coordinates. And so this was an excellent starting point. There were thousands of these observations that we saw across the United States. And so that was the plan, is that we were going to use this and then rely on, so as part of the grant, the idea was that we would be able to work with citizen scientists, not only to identify these populations, but in theory to help with collections. So we would be able to have a larger geographic distribution, and then we'd be able to supply a monetary honorarium for their efforts, as well as a lot of these folks are very interested in biodiversity conservation. So knowing that they're being a part of like saving these seeds for the USDA seed bank. So after we had a big collection of sites, these are 2 postdocs from my lab, Dr. Ademola Ina, Dr. Pawan Basnet. And so Ademola is kind of leading this project. And what he did is he had to filter through these sites because, as you might suspect, some of these are not true feral hemp. So there are different ways that we use different screening techniques, a lot of them just looking through images to better understand, like, does this appear to be in a site that was likely an escaped population? You know, is it in primary farm land? Is it in someone's backyard in a pot? There were lots of things that we used, the time of the observation, as well as just locations, like if it was in a rural or urban environment. So filtering through all of those sites for potential true feral hemp sites was the first thing that we did. And then we have reached out to collaborators. There are many people across the country that we work with that have agreed to make collections for us in specific regions, as well as some of the people and iNaturalists have agreed to help with collections. So we then, we have a federal USDA hemp license for research, in which that we've used to make lots of collections, and then have gone to these sites to verify whether or not they were true feral hemp sites. We normally will seek permission from the owner of the property if it's on private land, or a lot of these tends to be in state or state parks or along rivers. And then, yeah, go verify that it looks like it is feral hemp and that is not grown purposefully, and begin collection. So going through the collection protocol, the main aim here is to collect as much seed as possible, but also to make them very diverse, genetically distinct populations. So we encourage collectors to look for populations that have at least 10 female plants, but ideally more than 50 female plants. And this is typically happening in late September, early October, or I should just say in September and October when these collections are being made. So sometimes people will go out earlier in the year to scout if a population exists. And then if they're not comfortable, Adamola and folks from Dr. Weiblin's lab have driven all over the Midwest making these collections in September. And they will go out. And one thing that's very important is we focus on populations that tend to be at least 5 miles away from each other. So hemp being this wind pollinated, or cannabis being a wind pollinated species, you know, the pollen has the possibility to transfer or flow for very long distances. So we want to try to have discrete, isolated populations. So we're looking for these populations, like, you know, that they're not just continuous along an interstate or a highway. Then we've come up with this collection code that really just helps us for tracking purposes of where the state was collected and what population and what plant number. It's very important that we collect the GPS coordinates from these sites, so potentially we can check over time what is happening to these populations, as well as if future collections need to be made. And then we normally have collectors or ourselves record a site description of what's kind of going on in this area, and yeah, other notable features, and especially photos. So you can see one example of what this might look like. So once the populations are identified and they seem adequate, then it's just as simple as cutting the plants down up to 50 females. We right now keep all female plants separate. So we've been analyzing or collecting things on a plant by plant basis within a population. Everything gets its own little label. We do normally ask people to collect plant height just to have some metric, but the burden, I don't want to put too much burden on the collectors. So, and there are many reasons why any sort of trait data collected in those environments tend to be very environment specific. So that we just ask for height right now. And then plants are collected into individual bags. And then this is very important. So to make sure we normally want to have some leaf tissue and some seed tissue so we can preserve the seeds as well as start to get an idea of the cannabinoid profile. So we then ask people typically to dry the seeds, but if there's no way of them being able to kind of maintain a temperature less than 95 degrees Fahrenheit, we advise people not to do that, just because you're at risk of killing the seeds, which would be very sad to make the collection and then not have any seed left at the end of the day. But what works very well is just putting these under just a fan for a couple days, open in the brown paper bag. And then we just request people to hand strip the branches. You can condense the bag. And then, of course, putting the very important label, dried plant material for scientific research, not hazardous and of no commercial value. And then they get shipped to us, and we will provide all of the shipping labels and cost of shipping, and of course the $150 honorarium for each population that people help with. So getting into this, let's see what we've done so far. So these are some of the collections that we've made to date. We have over 1,500 individual plants. I'm sure it's well above this number now, actually. Across 10 states, we recently have obtained new collections from Colorado and New York. So that's exciting to add that beyond the Midwest. Of course, most of the samples being made around Wisconsin because that's what we know and easy to access, but definitely have been expanding more and more. And as the project people become more aware of the project, we get collectors from new regions, which is very exciting. So all these populations are logged into iNaturalist, so you can actually go and see some of the collections and the sites, and you can go check them out for yourself if you're ever so interested. Some of the things along with developing the protocol, when we first started doing this, reaching out to folks on iNaturalist, became very apparent that you can't just like contact someone and say, hey, tell me about that feral hemp that you saw. So really investing in the iNaturalist project and linking to our website and the project itself, and really showing that we're active part of the community has been very important to form those collaborations. One other thing is initially we had relied very heavily on hemp research and extension specialists at universities to help with collections because they already had a hemp license. But the last thing anybody who is doing work in hemp in September wants to do is anything else besides work on harvesting their own hemp. So we've actually learned that probably not working necessarily with people who are doing stuff in hemp specifically helps with making the collections. Of course, having the honorarium has been very helpful. It's not like people are doing it necessarily to get paid, but it can certainly help with the transportation costs and making sure people's time is valued. Just the protocol, we have a very clear protocol we send to people that's very detailed, that has resulted in people following it well. I have treat seeds with care just because the first year of collections, we had some seeds that went into a dryer, and the dryer went up to like 150 Fahrenheit and killed all of the seeds from a couple of populations. So just making sure that the seeds are not in, high humidity or high temperatures, which has very important. And that, of course, cannabis is still taboo, and that, this project is great. I love talking to people about it and talking about the differences between drug type cannabis and hemp, and the neat origin stories of where these feral hemp populations came from. So it's been really nice way to talk to people about hemp. So what do we do with all of those seeds that we've collected so far? So we wanted to understand the cannabinoid profile of them. So just a little bit of background. So when I talk about chemotype, so that's like the chemical profile type of the cannabis plant. We're just going to focus on these three types right now. So if you produce more THC than CBD, this typically is a type 1 plant. If you have equal parts of THC and CBD, this is typically a 1-to-1 ratio or equal ratio. And if you have less THC than CBD or increased CBD, that's a type 3 plant. And so there are these different classifications based on the composition of those CBD and THC molecules, and that relates to the genotype or the genetic control of those traits. So there have been molecular assays to be able to extract DNA from a plant and be able to determine if it's more likely to be type 1, type 2, or type 3 based on the genetics of the plant. So several of these assays have been developed that have scaled up kind of throughput. So there is this PACE assay that was developed, and really this relies on fluorescent tags that are able to distinguish the different types. So we have decided to use this PACE assay. So we extracted DNA from over 1,000 of these feral samples to better understand, do they have, what is happening at the genetic level? What is the likelihood that they're going to be producing more THC or more CBD or an intermediate amount? So we use this PACE assay that was developed out of the Cornell group, and then ran these on our samples. So just some preliminary data here to show you. There's a lot more data than this right now, but I think this just gives kind of a good snapshot. So if you look at the color here for the different types, so if you have this is kind of backwards, type 1 was the THC type, type 2 is the intermediate, and type 3 is the CBD type. So this is across different populations, and some of these are pooled by state and some of them are discrete populations. But the idea was to look at, okay, of all those plants that were sampled, what's happening? Is there a likelihood that they're going to produce a lot of THC? And this is another way for us to test, are they true feral samples? Maybe some of them were clandestine grows that were happening, that we're sampling. You can't tell sometimes under the density what the plants look like. And lo and behold, what we found is for the majority of the populations that we sample, they are true type 3s, meaning that they are not likely to make a high amount of THC. Although you can certainly see there are a couple of populations where we have a lot of the type 1 and type 2 plants in relatively high frequency. And really what this is providing us with information, when we're working with Zach at the seed bank to be like, okay, be cautious, these populations seem to have a lot of type 1 plants, so probably they're not going to be ready for prime time for redistribution. So maybe we can do some selections with them to really start to understand which seeds can be redistributed. And this is also important, and we're doing this on a per plant basis. So that helps. So if there are plants that are type 3, we know that we can likely use those. And so overall, you see that approximately 82% of the plants that we've sampled are of that type 3 nature, which should then be compliant. All these plants are also being subject to GCMS for true cannabinoid testing, and then looking at correlations between these chemotypic assays and the cannabinoid profile. And then finally, the idea, right, is to get these seeds into the hands of breeders and researchers so we can use them. So we want to reduce redundancy. There's all these plants. Are they unique? Do they offer different benefits? So the idea is to better understand, okay, of all these plants that we're collecting, these different populations, how similar are they to each other? Because it takes a lot of work to regenerate cannabis. It being dioecious, being wind pollinated, the way that you have to control the environment to only allow certain plants to cross-pollinate with each other takes a lot of resources. So we don't want to necessarily maintain 1,500 different feral hemp populations if they're not really offering that many unique genetic features or variation. So we started by taking a subset of these and sequencing them. And without getting into too much of this, the idea is really to use the genetic information to figure out how related they are to each other. So preliminary results from this. So I'm not going to go into a ton of detail here, but really the answer is that we're finding that there are these different subpopulations of the feral hemp that we have sampled. And most evidence right now is pointing to there being kind of 3 discrete / overlapping subpopulations. And that just means that these populations, so if we look at this plot right here, an individual vertical line represents a sample. And it's pretty much using the genetic information, we can assign an individual to a group. So it's looking at the genetic relatedness. And we see that a lot of populations, or a lot of these individuals, are clustering in this blue group, and then we have this red group and this K group. And using a statistical model, we're able to figure out that 3 groups best explains the variation that we're seeing. And when we look at this and overlay the geographic information, we see that this kind of clusters in these geographic groups. So if I overlay this with a map of the Midwest, because not everyone's from the Midwest, and I hate to say that we're a flyover state, but that's what we're called. But yeah, this is what the Midwest looks like. So just to refresh everyone, what we see here then is that there seems to be kind of a geographic distribution of these groups, where samples coming from where we collected further west in Nebraska and Iowa are having more genetic relatedness. Samples from kind of the center here in Wisconsin and in Illinois have a relatedness, and then Indiana. So we have a geographic distribution that we're observing. And that's exactly what you might start to think if there's different climatic features or geographic like there's differences in these locations. It could be precipitation or temperature or soil features that you start to see regional adaptation. So this is very promising that there are differences, and then we can use this to start to pick lines that really pull out the largest genetic differences. So we have priorities of what needs to be regenerated and maintained in the seed bank. So what comes next? So we still are sorting through all of our 2023 collections. So pretty much the data I'm showing you is from the 2022 collections. So all of the 2023 samples we've now also done genotyping on, and they're being genotyped, as well as the cannabinoid profile. So the goal is to bring this all together. And then ideally, this seed will be driven, because I'm not shipping it in the mail, to Geneva this fall, and should be available, in the very near future. Some of these feral hemp lines have already been distributed to the USDA seed bank. So if you go to GRIN, you can see some of these lines here from early collections. If you can't wait to kickstart your breeding program. So very exciting. Stay tuned. I hope this project will, at least the initial part of the project, will get wrapped up this year, but it's been very exciting. Next steps, or what happens after this? We have seed. So is this the end of it? I'm going to retire this year. No, there's lots of really cool stuff you can do with these seeds. So one thing it was mentioned, I'm very interested in the process of domestication. So fertilization, that kind of de-domestication, is really interesting. And without looking at this figure here, some work recently, Patrick Woods out of John McKay's lab, they looked at what you can do is look at the genome, and you can look for regions of differentiation between like cultivated hemp and hemp that has escaped or become fertilized. And there are these signatures of the genome that you can start to pick up on then, that mean there's something in that part of the genome that was important to be selected for when that plant escaped cultivation. So you can also reverse engineer that and think, oh, that might be really important for domestication as well. So you can start to look for these unique features across the genome and better understand what regions of the genome are important as the plant becomes fertilized or domesticated. So that kind of lets you get into potential places where there are genes controlling adaptation or domestication. So that's something I'm very interested in pursuing with all of our feral materials. I'm also very interested in just better understanding where these feral hemp lines came from. So we knew there was this introduction of these fiber types at different time points throughout history, right? So it was introduced into New England. We also know that then the initial USDA breeding program with Lester Dewey in the turn of the 20th century, that there was hemp that was likely Italian of it came from northern Italy that was then brought to Washington, D.C. They shipped that to Minnesota, where they had a breeding program. And then there were also introgressions into Kentucky. So I love the idea of, if we're seeing these different populations that are distinguished in the feral hemp, that it might be because they came from different sources. So here is just a mess that you don't really need to look at, but if you look, this phylogeny or tree is sorting out different types of hemp, and this just includes Wisconsin feral from a long time ago, but you can see kind of that it clusters with the Italian fiber types, but Chinese fiber types tend to be in a very different region. So I'm hoping as we start to include different feral types from across the country, we'll start to see subclustering and potentially figure out the origins of some of these. I love this little cutout right here from Lester Dewey. He had a little publication, Breeding Fiber Plants. I have it in my hands right now. But there's some really interesting stuff about this, of the origin of these different types of hemp. Normally they're coming from, like, they're named according to where they came from. So there's Chinese hemp, Japanese hemp, Russian hemp, American or Kentucky hemp. And then they talk about the different lines that have been developed. And a lot of the times the original US hemp cultivars are kind of these glued together geographic origins. So like Kymington was hemp that was coming from Kentucky, that then was bred in Minnesota, but was provided by seed from Lester Dewey from Washington. Or you can look at like Farmington. This comes from the Italian Ferrara cultivar, and similar, then crossed to Minnesota 8. And Chinnington being hemp coming from China that was then crossed to the Washington line. So a lot of these origins, they have some sort of linguistic information. And so it'll be really nice to match that with the genetic information to figure out where they all came from. So we're also very interested in using these lines in pre-breeding. So the goal this summer is to grow everything up alongside itself in a big common garden and start to collect data on these feral hemp lines for different things related to flowering time, plant architecture. I'm very interested in domestication traits, so things like related to dormancy and seed shattering and growth habits. So we will be growing those all. So I also invite you to come to the Madison field day on August 20th will be our field day at just north of Madison in Arlington, Wisconsin. So reach out if you're interested in that, to see all of these lines grown side by side, or all of these accessions. Just some preliminary data on that. So we've done this in previous years at very small scale. So part of the Midwestern Hemp Research Collaborative, we have joint trials across 4 Midwestern states. This is just to show that there is variation, right? So we put them in some of our cultivar trials. So this is for flowering time, so earliest flowering to latest flowering, different varieties here, or different cultivars shown, and you can see like the lines that we've used, the Wisconsin feral types, they have this very intermediate flowering time, so they're well adapted in our region. The flowering time works out well. We have that also for fiber yield. So here we have dry matter pounds per acre, and interestingly, of the 4 different feral hemp lines we put in there, we had 2 kind of low performers, but 2 that produce quite a bit of dry fiber biomass. So, it's interesting to see what potential they might have in fiber breeding programs. And then finally, I have a postdoc, Derek Grunwald, who has been doing a large survey of viruses across the Midwest and other hemp diseases. And we found some really interesting differences with not really market type, but if you look at feral hemp versus grain and fiber and high cannabinoid hemp, that for some specific viruses, feral hemp has a significantly less abundance of those viruses, and also a lower rate of co-infection of viruses. So just like kind of showcasing that there are some potential adaptive alleles that are persisting in those environments that might be useful for things like disease resistance. So with that, I'll wrap it up and say that So far for our collection, we're pleased that I think a lot of the stuff that we sampled is actually feral hemp, and that more than 80% of it right now is that type 3 chemotype, so it is compliant and can be redistributed pretty readily. We do see that there is different genetic differentiation between even our first year collection, where we have at least 3 subpopulations that we're observing. So this material, I think, is going to be a useful source of genetic variation for future breeding programs. And of course, cannabis offers opportunities. We know there's been lots of ups and downs, and I think there will continue to be, but nonetheless, that's true of everything. So I think that there's really still lots of opportunities to explore, both from providing profitability to farmers and processors and new jobs, as well as potential environmental sustainability with developing new cropping systems and integrating more genetic diversity into our current agricultural systems. So with that, I will acknowledge my other team members here. So at the top, we've got the Minnesota group, Dr. Weiblin, as well as JP Wenger and Joe P. Smeji, who have been very excellent colleagues and gone collecting on feral hemp and lots of fun. Dr. Adam Molina, who has done the majority of the collections, the postdoc in my lab, as well as my team and all the other citizen scientists or iNaturalist and members of the S1084 hemp hatch, multi-state hatch program that have helped with collections. And this has been funded by USDA's Supplemental and Alternative Crop Grant. And I will leave it with this. We're not really collecting more ourselves this year, but the project will go on. So if you know where feral hemp is, get in touch with me, scan that. At the bottom of that page, you'll find more information, but we'll still happily collaborate with you to collect more samples. And there's also our extension website and my lab website there. And I will stop at that. Thank you. All right, let me look through these here. Do scientists know for certain cannabis did not exist? Okay. Yeah, so question about, do we know for sure that cannabis did not exist in North America prior to colonization? So I think there's pretty concrete evidence that it was not here prior to colonization. I don't think I have more to say about that. I can't like tell you how I know that for sure, but I don't think there's evidence that it was here. And definitely the center of origin and domestication being more in Central Asia, and then kind of the spread, I think, has been well studied. So I'm happy to point to some of that literature, but I think so. How can state seed certification agencies be involved in Germplasm Project feral hemp efforts? State Seed Certification Association Germplasm Project. Yeah, so one thing I can just mention is, you know, it's been difficult. A lot of the times the collaborators, the citizen scientists, it's very helpful for us just for people to make the initial identifications of the populations. I completely understand it is very worrisome to people to be like, okay, no, you're part of this project. Here's this research license. Go climb into this like pasture and collect this feral hemp. It's totally legal. So even just the verification of population. So once something has been found on iNaturalist, having someone go to get a second pair of eyes on it to verify that it's still there means the world for us when we're like planning our collection trips, because yeah, I mean, probably 50% of the time we'll go to a site and there's nothing there, which is very disheartening. So that's a great place that people have been very helpful, is even just verifying potential spots. And as far as, you know, just spreading the word for this project is very helpful and reaching out. So the more reach we have, the better. So question about farm fields abandoned by farmers. So one interesting thing, and I think was very topical, was we have, so we have a little bit of time. So we're growing hemp now, right? We didn't grow hemp for many years. I think the last hemp field was harvested in 1958. There was a tiny little hemp field in Wisconsin still. So there was no hemp. So then when I started looking at this and you're making observations of hemp in 2018, 2019, you're thinking, okay, that could be feral hemp, right? Because we didn't have our federally legal program yet, but now we're growing a lot more hemp. So there's a chance now that there's actually recent escapes into cultivation, or people have just left fields abandoned because they didn't end up harvesting them. So how do we differentiate from that? And that's a big problem. So I think we're in a really important time point where we need to be making sure we're capturing these feral populations before we see more gene flow between the new cultivated populations or just new escapes, because the goal is not to be collecting this new material or material that is under any sort of IP. So I do think that there's a bit of a time crunch. You can also, communication goes a long ways. You can know if hemp has been grown in that area recently, as well as a lot of the times where we're collecting these, they're not in cultivated landscapes. They're, like I said, in pastures or along train tracks, along riverbeds. So it's not likely that someone was growing there. So of course, people are asking about collections. So hopefully this screen in front of you can help with that. There's a question about monoecious feral hemp. So when we're making these collections, we only have really like one shot when we're doing a big trip. So this tends to be, as I mentioned, in late September, we'll do these trips. And by that time, normally all the male hemp is dead. So we tend to only see mature female plants because we want the grain or with the seed to be ready. So I have seen feral populations we've replanted in the greenhouse have monaceae where you will see a few of them. It's very, very, very low frequency so far, though. Most of the populations seem to be dioecious, but there is that bias that when I'm collecting, I'm only really seeing female flowers at that point. But I have seen a very low frequency of it in the greenhouse. So viruses, so the virus survey work will be coming out. We've just been testing for we've tested for 6 or 7 different viruses that I'm happy to share that data. There's cannabis sativa mitovirus and tobacco streak virus and AMV and yeah, without having that data in front of me, I can't really tell you off the top of my head the results, other than that we are seeing differentiation between the feral types and the cultivated types. All right, how can we get involved? Yeah, I think a lot of these questions have to do with people getting involved. So certainly, please email me if you're interested in making collections. It would be excellent to have more of this. I'm sure Zach would be happy to have more, and I know people are interested in getting their hands on a lot of the feral seed, and I really hope it will be a useful resource. And then the last question, phenotypes that are most promising to integress into commercial backgrounds. So I think we haven't even begun to scratch the surface yet about what these plants will look like when we collect them. You can collect a feral hemp population that's like on the side of a chicken coop, and it has like a bunch of manure, and it will be like 14 feet tall and look amazing, and then you grow it in the greenhouse and it's a tiny little thing. So I think the common garden is going to be really important as we start to figure out what's going on here, getting them growing all in the same place and starting to take much more detailed phenotypic notes. And then I think stressing them, I think the abiotic and biotic stress resistance to, things that maybe are going to be related to disease or pest pressure or competition or resource management are going to be where they're going to thrive. So that's where I'm hopeful for. Yeah, I don't know when to end. I'll answer this last question. So if you're collecting in September, are you missing things that mature earlier? So yes, more than likely. And especially, I've had people reach out from places like Oklahoma, and I don't know exactly when the plants are maturing. One thing that is helpful is on iNaturalist, people will tell you like when they spotted it, and you can kind of look at maturity based on when they saw it and what the pictures look like. And because of the indeterminate growth habit of cannabis, it will start shattering seed, and there will be you will lose seed, so you have to time it correctly that there's enough mature seed, but it's not all shattered. So you kind of have like this 2 or 3 week window to make collections. But yeah, it's definitely going to range by latitude. But yeah, that's why it's really important to get as many sets of eyes on the populations as possible. So even if one year you're like, oh, I missed it, then that helps with knowing next year you need to go earlier.
Okay, thank you, Shelby, for closing out our series and answering all these questions. We really appreciate you sharing the work you're leading on capturing the genetic diversity we have in our feral germplasm here. Also, since we're closing out the series, I want to give a big thank you to Craig Kramer, Kim Paul, and Malin Murdoch for helping me with the logistics and advertising and media for this webinar series. And thank you to all of you guys for joining us. Hope to see some of you at our field days in September at Cornell, and as Shelby was saying, in August at Wisconsin. And also next year virtually at our 2025 hemp webinar series. Also, best wishes for all of our cultivators out there in the upcoming field season. And I'll let you all enjoy the rest of your day. Thank you again, Shelby.
Thank you.
This has been a production of Cornell University, on the web at cornell.edu.